Luminex's xMAP technology makes up the basis of Luminex's core business. This flexible technology is centered on the use of Luminex xMAP microspheres.
xMAP was originally an acronym, meaning multi-analyte profiling, where you solve for X. In other words, analyzing data from an assay, checking for multiple analytes in a single test.
The surface of the microsphere is covered with carboxyl groups, which offer multiple chemical attachment sites for biological capture molecules. The attachment of molecules to the microsphere is called coupling. Carboxyl groups on the bead are reactive, allowing bonding to both nucleic acids and proteins. Because carboxyl groups are so reactive, it is a versatile and easy-to-use chemistry. With the bead coupled to the desired capture molecule, it can now bind with a specific target. Using a reporter molecule with the target makes identification of that target possible.
For example, beads 12, 35, and 42 have specific dye concentrations that are detected by Luminex instruments and used to identify each bead. Additionally, each bead has a single target it can bind with, which is determined by the capture molecule that is attached to the bead. For example, either X, Y, or Z, respectively. So if target X is present in the patient sample, B12 will bind to the target. The target is then bound to a reporter molecule that the Luminex instrument also detects, associating it with B12 and identifying that the patient has disease X. The other beads are still red, but since targets Y and Z are not present, no reporter will be detected on those beads. That is how xMAP technology works.
The analytes of interest are commonly nucleic acids or proteins. Following are examples of specific uses for nucleic acids and proteins.
Nucleic acids, particularly oligonucleotides, are common analytes of interest that can be coupled to microspheres. A nucleic acid sequence specific to the target is attached to the carboxyl group using an amine linker. The amine linker positions the capture probe away from the microsphere's surface and facilitates binding to the target. When the sample is added to this reaction, the nucleic acid sequence on the bead binds to its complementary nucleic acid in the sample, if present. This is called hybridization. Biotin is incorporated into the target to provide a binding site for the reporter molecule.
Proteins are another common analyte of interest that can be coupled to microspheres via the primary amine groups naturally present in proteins. Coupled proteins can be used as capture molecules to bind specific protein targets. Protein targets can be directly or indirectly identified via binding interactions with capture proteins attached to the microspheres. These binding reactions occur between various proteins, including antibodies, antigens, receptors, ligands, and enzymes.
Luminex currently has multiple xMAP instruments based on either flow cytometry or fluorescent imaging detection technologies. Luminex's FlexMap 3D, Luminex 200, and xMAP IntelliFlex instruments use xMAP flow cytometry-based technology to identify beads and if a target is present. With xMAP flow cytometry, the beads are fed into a compartment called a cuvette and individually interrogated by lasers. The red laser excites dyes within the beads to identify the bead region. The red laser works with a set of sensors that detect the dye concentrations of the bead. The green laser identifies the fluorescence of the reporter molecule. It works with its own sensor. Both the bead ID is mapped and the median fluorescence intensity is measured. In addition, the xMAP IntelliFlex portfolio has a model with a third violet laser, which enables workflows that include detection of a second reporter. This is useful for applications such as detecting multiple targets on the same bead. The Luminex MagPix instrument uses xMAP fluorescent imaging analysis. This method reads all the beads at once. The beads are fed into an imaging chamber where they are captured by a magnetized wall. The magnetic MagPlex beads are required for this method. Once the beads are in place, red and green LEDs shine on the beads to excite the dyes and reporter. Then, a camera is used to identify each bead by region as well as quantify the analyte binding events. Once complete, the magnet moves away from the chamber, releasing the beads to be flushed from the instrument.
Luminex xMAP technology is based on the use of Luminex microspheres. The reactive surface chemistry of the microspheres provides a flexible platform for the attachment and detection of multiple types of biological molecules. Luminex's wide range of xMAP products allows for a customizable end-user experience.